Hello, my name is Henry. I'm a rising sophomore at Johns Hopkins University, and this summer I was able to work with the Gun Lab here at BTI in Cornell. My project is looking at rubisco. Rubisco is the limiting enzyme within photosynthesis, and it's basically very slow and inefficient. And our lab looks to engineer it to have faster kinetics, the speed of which the reaction takes place. So we take a look at the small subunit. Now, rubisco has 8 large and 8 small subunits, and the small subunits are interesting because they're known to influence the speed of which the reaction. However, they're very distant from where the actual reaction takes place. So Maddie, a member of my lab, made an energetic landscape, which is basically a picture of all the possible mutations in both the large and the small subunit, and showed them how destabilizing they would be, or how destabilizing and how stabilizing they would be. We found 6 outliers for further experimental validation, and we basically hypothesized that destabilizing mutations would increase the speed at which this reaction takes place. You can kind of think about it as WD-40 on a door hinge. You're kind of loosening this enzyme, and it allows it to react much faster with its substrate. So for our results, we found that rubisco with stabilized mutations actually were successfully assembled. We had very little changes, and LASA expression was great. For rubiscos with destabilized mutations, however, we showed very little expression, and this was probably due to the mutation itself interfering with the folding of the small subunit, and it's preventing it from folding correctly because rubisco is a very complicated enzyme and needs chaperones and chaperonins, which are basically its support system to fold correctly. Now, our most interesting result is that the stabilized mutations had significantly decreased reaction speed, which is amazing because that means we can start using computational tools to engineer rubiscos with desired reaction rates. Moving forward, we can think about implementing these mutations within live plants to see how those plants look in terms of a lab environment versus in an actual growth chamber. And ultimately, I would just like to thank my lab for supporting me this summer. It's been truly amazing.
You did a great job. I really liked your analogies in your talk. What was the biggest challenge in this project as a whole?
The biggest challenge had to be the PCR reactions that I did. They just were not working for no reason. And then they finally worked. It's a mystery.
Well, I'm sure your skills improved so that you could get them to work, but really nice poster. Great job.
Thank you so much.